Hello everyone. Continuation of the previous video, we had seen in the last video about the one of the technique of thermal processing, that is the pasteurization technique. So coming to the sterilization technique, so we all from the term sterilization we can come to a conclusion that it is like completely all the bacteria, that is even the spore forming bacteria, are to be going to get sterilized. Okay, so keeping that in mind, what we have to look in that sterilization is nothing but the process that involves complete heating is being taking into role, and in that place what happens is that either by using a dry heat or moist heat method, or even by filtration, other methods, we can undergo the sterilization process. Okay, so this sterilization technique is actually much adopted one in most of all the FMCG industries, because only when any product is sterilized, we can actually like confidently get the recognition, and we can go in for export purposes also. Okay, so coming into the sterilization, what happens is actually how the food will be sterilized is we have an equipment called sterilizer. So the role of sterilizer is nothing but to sterilize all the food materials that are being going to kept inside an sterilizer to 100 degrees Celsius. So this will be a comes under a mild process. This will comes under a severe process of heat treatment only, in order to preserve any food material. Okay, so food products will be like kept filled, or it will be in a sealed condition only inside a sterilizer. So mainly the temperature will be ranging compulsorily 100 degree, but sometimes based on the product the temperature will range from 110 degrees Celsius to 121 degrees Celsius. So what happens is that the heat will get the heat must be we have to make sure it reaches the product inside. Okay, so in such a way here also it will be dependent on the time and the temperature that we are going to use for sterilization technique. Okay, so mainly the time temperature will depend upon the size of the container as well as what type of product we are going to use for sterilization. Okay, so how this process will be carried out is actually in this process there are 3 phases, or the 3 stages will be done. Okay, so what happens is that phase we can conclude like one is the phase 1, phase 2 and phase 3. So what happens in the phase 1 is nothing but we call it as the first, when a product is exposed inside a sterilizer, first it gets heated, then holding phase, and then cooling phase we can call it. Okay, so for heating phase, what happens in heating phase is here the medium of heat that passes into a sterilizer will be either water or steam, and it will be like based on the ambient temperature. Based on that product that we are going to keep inside, the temperature will start to increase. Okay, in phase 2 what happens is that in holding phase, that particular temperature that it has reached will actually maintained for some defined time in order to kill all the spore forming bacteria that are present in the food product. Okay, and last we have to undergo the phase 3, that is nothing but the cooling phase, that it has to be like we have to reduce the temperature that has been maintained or that has gone inside the particular sealed container or product. By maintaining it in autoclave condition, we have to cool down or reduce the temperature inside the particular product, and then only we have to take it out of the sterilizer. So this is in the diagram we can see this is the sterilizer equipment. So otherwise we call it as like those equipments are called as a sterilizer, or sometimes we call it as the autoclave or the retorts. So mostly in an industry we term it as, in our terms we call it as autoclaves. So what happens is that the thermal treatment has to be performed under the pressure. So mainly by creating a pressure inside an autoclave only, we actually go in for sterilizing a particular product. So it actually acts like a pressure cooker only. Okay, that only is the principle of sterilizer. So high temperatures will be generated by either we can use a technique called, we can either inject the steam, or we can actually by existing water that will be along sides of the autoclave, it will actually start to boil and it will create a steam inside and then increase the temperature over 100 degrees Celsius, and by the combination of steam and water heating, a sterilization equipment will function. And then coming to the what happens, actually we are telling that the all the bacteria are going to get killed. So what are all the looking into the kinetics of the microorganisms, what happens? So mainly those organisms depend upon the like how based on what factors those organisms will get killed is the that bacteria cell's age, then initial concentration of the cell or the spore, and the medium in which that bacteria has grown, the moisture content of the particular dry food, and the pH of the food. So these are the 5 different types of factors that will contribute to the microorganisms resistant to the sterilization technique. Thank you.